The bones were not supposed to be there. When archaeologists working beneath the foundations of a Sumerian temple complex in southern Iraq broke through a sealed layer of compacted clay and reached a burial chamber that had not been opened in 7,000 years, the expectation was that what they would find would fit within the established framework of what we know about the ancient Near East: early agricultural communities, Neolithic burial practices. Skeletal remains consistent with the populations that archaeologists have documented across the Fertile Crescent from roughly the same period. The bones would be analyzed, catalogued, compared to the existing database of ancient human remains from the region, and the results would add detail to a picture that was already reasonably well understood. That is not what happened. What the DNA analysis returned when the samples from the sealed burial chamber were processed through the full suite of modern ancient DNA extraction and sequencing techniques was a result that the laboratory team ran 3 times before reporting it, because the first result seemed like contamination, and the second result seemed like instrument error, and the third result was identical to the first 2 and could no longer be attributed to either. What the bones contained was a genetic profile that does not fit within the established population history of the ancient Near East, that does not match any known ancient population from the region or from anywhere else in the world, with a clean and direct correspondence, and that contains within its structure a set of genomic features whose distribution across the human genome is not consistent with the patterns produced by normal population mixing, migration, and genetic drift over time. The scientists who produced this result are not fringe researchers. They are members of a team with institutional affiliations at multiple universities whose ancient DNA work on other populations has been published in peer-reviewed journals and has been accepted without controversy within the field. The result they produced from the Sumerian temple bones is not being disputed on methodological grounds. It is being debated on interpretive grounds, because what the data shows is clear enough. What it means is the question that is dividing the researchers who have reviewed it, and that is now moving through the broader scientific community in the way that genuinely anomalous findings move, slowly at first, and then very quickly, once the implications become impossible to contain within specialist conversation. To understand what makes this DNA result significant, you need to understand what ancient DNA analysis can and cannot tell you, and why the specific pattern found in the Sumerian temple bones stands out from everything else in the database of ancient human genomes. Ancient DNA analysis has transformed archaeology and human prehistory research over the past 15 years. The ability to extract and sequence genetic material from skeletal remains thousands of years old has allowed scientists to answer questions about human population history that no amount of traditional archaeological analysis could resolve. Where did the first farmers of Europe come from? How did the Indo-European languages spread across Eurasia? What was the relationship between the people who built Stonehenge and the people living in Britain today? These questions have been answered with a precision and a certainty that was not possible before ancient DNA became technically feasible at scale. The database of ancient human genomes now contains thousands of individuals from across the world and across time, from remains as old as 40,000 years to remains from just a few centuries ago. This database allows researchers to place any new ancient genome in its population context by comparing it to the full range of known ancient populations, and identifying which combinations of ancestral populations best explain the genetic profile of the individual being analyzed. Every ancient genome analyzed so far, including genomes from populations that were geographically isolated, genetically distinctive, and previously poorly understood, has fit into this framework in the sense that it can be modeled as some combination of ancestral populations that are themselves represented in the database. The Sumerian temple bones do not fit this framework in the expected way. They fit partially. The genomic analysis shows that a significant portion of the genetic ancestry of the individuals buried in the sealed chamber is consistent with the early Neolithic farming populations of the ancient Near East, the people who developed agriculture in the Fertile Crescent, and whose genetic signature is well documented across thousands of ancient genomes from the region. This component, roughly 60 to 65% of the genome depending on the individual, is not anomalous. It is exactly what you would expect from a population living in southern Mesopotamia 7,000 years ago, based on everything the existing database says about population history in that region. The remaining 35 to 40% is what the laboratory team ran 3 times. This component does not match any population in the ancient DNA database. It does not match populations from Africa, from Europe, from Central Asia, from South Asia, from East Asia, or from the Americas. It shows the closest affinity, when forced into the nearest neighbor analysis that geneticists use to identify the best available match for an unknown population, to a cluster of populations from the Caucasus Mountains region, specifically a component that is also found in lower proportions in ancient genomes from the Zagros Mountains of western Iran. But the match is not clean. The Sumerian temple genome component is related to these Caucasus and Zagros populations, but is not derived from them in the straightforward way that population mixture events produce. It is as if the Sumerian component and the Caucasus component share a common ancestor that is not currently in the database, an ancestral population that contributed genetic material to both, but that is not itself represented by any currently sequenced ancient genome. This kind of finding, a genomic ghost population, is not unprecedented in ancient DNA research. Geneticists have previously identified ghost populations, ancestral groups that left genetic traces in multiple living or ancient populations without themselves being directly represented in the fossil or ancient genome record. The most famous example is the Denisovan ghost, an archaic human population known primarily from a single finger bone and a few teeth, but whose genetic contribution to modern human populations across a wide geographic range is clear from the signatures they left in the genomes of their descendants. Ghost populations in ancient DNA are real, and their discovery is a legitimate and important part of the field. What makes the Sumerian temple ghost population different from previously identified ghost populations is the nature of the genomic features that distinguish it. When geneticists characterize a ghost population by its contribution to known genomes, they look at the pattern of genetic variants that trace to the ghost ancestry and ask what those variants tell them about the ghost population's history. In the case of Denisovan ancestry in modern humans, for example, the variants that trace to Denisovan ancestry are distributed across the genome in a pattern, consistent with a single admixture event in the distant past, followed by thousands of generations of normal human population evolution. The pattern is old and thoroughly mixed into the recipient population's genome in the way that time and recombination produce. The ghost population component in the Sumerian temple bones shows a different pattern. The variants that trace to the unknown ancestry are not distributed across the genome in a way that is consistent with a single ancient admixture event, followed by normal mixing over many generations. They are distributed in a pattern that has unusual structure, specifically a pattern of long genomic segments carrying the unknown ancestry that are longer than the expected segment lengths for ancestry introduced thousands of generations ago. Long segments of shared ancestry are preserved when admixture was recent because recombination has not yet had time to break the segments into smaller pieces. Short, fragmented segments indicate ancient admixture. The Sumerian temple bones show segment lengths that suggest the unknown ancestry was introduced not in the deep past, but relatively recently before the time these individuals lived, within a few hundred generations rather than thousands. This finding has a specific implication that is as uncomfortable as it is clear. If the unknown ancestry component in the Sumerian temple bones was introduced into the population relatively recently, before 7,000 years ago, it was introduced by a population that existed and was mixing with the local Mesopotamian population sometime between approximately 8,000 and 7,000 years ago. A population that has left no direct representation in the ancient genome database, despite living in or near the most archaeologically documented region in the world, during the period when humans were most intensively settling and developing the Fertile Crescent. A population that was present in Mesopotamia during the formative period of what would become Sumerian civilization, that contributed a substantial fraction of its genetic material to at least some members of that early Sumerian community, and that then disappeared from the record so completely that its bones have not previously been found and its genome has not previously been sequenced, is not an explanation that sits comfortably within the established narrative of how Sumerian civilization developed. The Sumerian civilization itself is already one of the most remarkable puzzles in human history. It appears in the archaeological record with a suddenness that has always required explanation. The transition from the Ubaid period, a relatively unremarkable Neolithic farming culture in southern Mesopotamia, to the early Sumerian civilization with its cities, its writing system, its complex theology, its sophisticated mathematics, its astronomical knowledge, and its unprecedented administrative organization, happens over a period that is short by the standards of cultural development. Civilizations of comparable complexity elsewhere in the world developed over longer periods from more visible cultural antecedents. The Sumerian emergence has always suggested either a very rapid internal development, a contribution from an external population with existing sophisticated knowledge, or some combination of both. The DNA finding adds a specific and testable dimension to this question. The ghost population whose genetic contribution appears in the Sumerian temple bones was a real biological population. It lived. It mixed with the local farming communities of early Mesopotamia. It contributed genetic material to the people buried beneath a Sumerian temple. And then it vanished from the archaeological record so completely that 7,000 years of subsequent excavation in the most archaeologically studied region on Earth had not previously turned up its bones. The location of the burial matters. These bones were not found in a domestic context. They were not found in a cemetery. They were found beneath a temple, in a sealed chamber, in a position suggesting deliberate and careful interment by people who understood these individuals to be significant enough to bury in the most sacred space available to them. In Sumerian and pre-Sumerian religious practice, burial beneath a temple foundation was reserved for individuals of the highest religious or social importance. The people buried in this chamber were not ordinary members of the community. They were individuals whose status was high enough to earn them a place in the physical foundation of the community's sacred space. The combination of the genomic anomaly and the burial context has led some of the researchers involved in the analysis to a hypothesis that they have stated carefully and with appropriate academic qualification. But that is clear in its direction. The individuals in the sealed chamber may represent members of the population that contributed the unknown genetic component to the early Sumerian community, individuals who were understood by the surrounding population as belonging to a distinct group of special significance, and who were honored in death by burial beneath the temple, in recognition of a status that the community recognized and preserved even as the genetic boundary between the two populations blurred through intermarriage over subsequent generations. This hypothesis does not require the invocation of anything outside normal human history. A population that arrived in Mesopotamia from an unknown origin, that possessed knowledge or capabilities that the local farming communities did not have, that was recognized as distinct and significant, that intermarried with the local population over several generations, until the genetic boundary between them became indistinct, and that was honored in death by the community that had absorbed them, is a normal human story. It has happened many times in documented history. What is not normal is the complete absence of this population from the existing archaeological and genetic record up to this point. The geographical origin of the ghost population is the question that the existing data cannot fully answer, but that it points toward in suggestive ways. The closest affinity to the unknown genomic component is in populations from the highland regions to the north and east of Mesopotamia, from the Zagros and Caucasus mountain zones. These are also the regions that ancient Sumerian texts describe as the homelands of specific groups of divine or semi-divine beings, the regions from which, according to Sumerian mythological tradition, certain forms of knowledge and certain special lineages descended into the lowland civilizations of the river valleys. The Sumerian tradition of the Anunnaki, the beings described as coming from the mountains or from above to establish civilization in the lowlands, has been interpreted in many ways. Theological interpretation sees them as genuine divine beings. Rationalist interpretation sees them as mythologized memories of real leaders or cultural innovators whose achievements were so significant that later generations elevated them to divine status in cultural memory. The genetic finding adds a third possibility that sits between these two, and that is grounded in biological evidence rather than textual interpretation. The Anunnaki of Sumerian tradition may preserve a cultural memory of a real distinct population that arrived in Mesopotamia, contributed knowledge and genetics to the developing civilization, and was subsequently remembered as divine because their origin was unknown, their capabilities were impressive, and their genetic distinctiveness marked them as visibly different from the surrounding population. This interpretation does not require the population to have been anything other than human. It requires only that they were a population whose geographic origin placed them outside the genetic network of Fertile Crescent farming communities, that they arrived in Mesopotamia during the formative period of Sumerian civilization, that they mixed with the local population over several generations, and that the memory of their arrival and their significance was preserved in the mythological tradition that later became the written Sumerian texts that archaeologists and historians have been studying since the 19th century. The DNA changes everything in the sense that it takes these questions out of the domain of mythology and cultural interpretation and places them in the domain of biology. Whatever the ghost population was, whatever it knew, wherever it came from, it was real. Its members had bodies. Their bodies had DNA. That DNA is sitting in a database now, sequenced and analyzed and waiting to be matched to other genomes that have not yet been found. The bones under the Sumerian temple have spoken in the only language that does not lie, the language of genetic sequence, and what they have said is that the story of where Sumerian civilization came from is not yet fully told. The sealed chamber has been opened. The bones have been sequenced. The result has been verified 3 times. And the question of who those people were, where they came from, and why they were buried in the foundation of a Sumerian temple, 7. 1,000 years ago is now the most interesting question in the ancient DNA field, and possibly the most interesting question in the study of human prehistory. The database will grow, more bones will be found, more sequences will be run, and the ghost population that left its genetic signature in the foundation of Sumerian civilization will eventually be identified with more precision than the current data allows. Until then, the bones speak. And what they say is that the history of human civilization has a chapter that has not yet been written, a chapter whose existence was unsuspected until a sealed clay chamber was opened, and 3 rounds of DNA sequencing returned the same anomalous result that nobody expected, and nobody is quite sure how to fully explain.